you. Wow, what a great treat, Ken. Thank you. Thank the Nutri group of professionals. It's so exciting for me to see the evolution of Nutri over the now nearly 30 years that I've had the privilege of being with my UK family. So it's really a great privilege to be here today. And I want to also acknowledge on behalf of my colleagues at the Personalized Lifestyle Medicine Institute our pleasure to be part of this kind of global consortium that's hopefully creating positive change in health in people in need across the globe. And I want to acknowledge my colleague and friend, Annette Giardi. Annette, if you'd stand up, who is here all the way from Gig Harbor, Bainbridge Island, Washington State, representing PLMI. Please stop by. She's give you information about PLMI. We have a very robust website. We have all of our programs that we have done over the last 7 years, all the videos of the international speakers that are available free of charge off of our website. And we've got a meeting coming up October 11th and 12th in Seattle. It's on personalized nutrition. I think it's going to be quite remarkable. And Annette also is a native of England. It turns out that she and her oldest sister and her daughter are here doing some touring. They're going out to where Annette grew up as a child, outside of London, so it's really neat to close her links with her family history here in the UK. For me, it's just a really, really great privilege once again to be here to kind of share some of the thoughts that we have been working on now for, when I say it, it almost scares me to say it, 4 decades. And I just had my 73rd birthday, and I'm thinking, man, oh man, this is kind of interesting how time does go by. And in fact, just to give you an anecdote, I have 3 sons, and 2 of them are married, and I have 5 grandchildren, 2 remarkable daughter-in-laws, both of who I would have chosen as women for my sons to spend their life with. I was one of those lucky fathers that really got great daughter-in-laws and great mothers of our grandchildren. And so I was talking to my oldest son, who's just turning 50, and I was kind of trying to give him some fatherly advice, and I was recognizing, you have to be very sensitive. You can't tell a 50-year-old son what to do, but I was trying to guide him, and he was being very polite and pleasant with me, and then he says, "Dad, thanks a million, I am 50. Yeah, so it reminded me, and then he went on to say that he's experiencing middle age, and I said, don't even go there with me, well, let's not go down that path. But one thing I can say is over 40 years that I've had really remarkable experiences in meeting some tremendous leaders in healthcare around the world, and my flight travel plans have taken me in places I never would have expected. I've done well over 6 million miles of air travel over the last 40 years. And I've had the chance to hang out with a lot of people like yourselves that are really visionary, warriors, courageous, people that are, sometimes on the road less traveled, a little bit more difficult than staying on the traditional path, but doing so with a great commitment to service and to assisting people who are less fortunate, who have needs, and trying to find solutions to complex problems that relate to health, which cover a whole spectrum of things, as we know. Health is a term that you can either have a small case L or H, or you can have a big case H. And the big case H, the capital H health, is really that encompasses everything that the human experience brings. The world in which we live, the plants and animals, the ecology, the nature of our relationships, our spiritual belief systems, how we eat, how we believe, think, act, what we drink, how we move. All these things are part of the big capital H. And so when you take on this field to make your life's purpose, you're taking a big bite, and it's a big appetite to hold on to. So I felt really privileged to be a part of this community, and I don't think I've really, in those 40 years, come up with a lot of original ideas. But I think I am a very good listener, and I'm a mosaic of all these experiences that I've had, and I think I can put them together in ways, hopefully, that make sense and motivate other people to take the baton and to move on with how they would like to make their service available. So that's what I'm going to be sharing with you this morning, some thoughts about that journey, and hopefully some of this will relate directly to how you can better serve your clients or your world that you're involved with. So let me start with the concept of personalization, because we're moving in an interesting time in history from what I would call the age of being part of the average, being part of the group, being part of the community. When I was born, I was the first of the baby boomer years. I was born in 1946, and this was right after World War II, and at least in America, and I would expect here in Britain as well, people wanted to get over the war mentality. They wanted to be part of the group. They wanted to be resurrecting culture out of the ravages of World War II. And it was kind of a cornucopian time in which the objectives were to have a house, have a TV, have a car, and have a safe and peaceful life and be part of the community. So the concept was to be part of the group. And in fact, that's to a great extent the way medicine grew up. Medicine grew up with the concept of what I would call population biology, epidemiology, statistics, Gaussian curves, looking at large numbers of people and then asking what is the average person like and how can we service that average person. So when you then go to a disease, you have a Gaussian curve of the disease. People that they say have diabetes, not everybody with diabetes is the same, so they present with a various types of severity and duration and frequency of symptoms. So they form a distribution curve, but you treat the middle of the curve because we're into the rule of the averages. Does this sound familiar at all? And that's how medicine kind of grew up as part of a culture over the last 70 years. And there's nothing wrong with that, by the way. And it served great purpose, and we can see that the advances that were made in the treatment of disease that were indications of this kind of coalescence of different signs and symptoms that gave rise to our understanding supposedly of histopathology as to what gave rise to the name of that disease served very well to build what we call hospital-based medicine, which really has been very successful in treating end-stage disease. However, now we're moving culturally into a different time. And there are many forces that have created that cultural change, which I won't go into the sociology, but certainly social media and the internet is a big player that allows people to move from thinking themselves as a member of the group to thinking themselves as an individual that they select to be their tribe or their affinity group. So now we have different kinds of music, different kinds of art, different types of literature, different types of going back and studying your ethnic roots and affiliating with those in an individual way so that we're moving from the age of the general community population to the age of the individual. And that's a global trend that's occurring everywhere I go, where we're seeing the resurrection of the importance of the individual. Now there's some puts and takes to that because you can take anything to the extreme and you might say, well, does that make us then too self-centered and make us too kind of, it's all about me type of thing? There is some of that, but I think what it also has done, the positive, is it's created an individual to express their uniqueness on the planet. If we're all unique facets on the diamond of life, let me say it again. If we're all unique facets on the diamond of life, carrying unique genes that make our book of life uniquely different than anyone that's ever been born before or anyone that will ever be born after. We're a unique chapter in the sea of humans. And so with that as a construct, it is really kind of changing all sorts of institutional structures, one of which is medicine. And that's obviously where I'm going to be speaking, because I'm going to be speaking about the nutrition connection to individualization and personalization. That is a revolutionary change, a force of change. It's not just here today and gone tomorrow. It is a real force of change that's changing all aspects of healthcare delivery. And I'll touch upon some of those over the course of the next few minutes. But let's start, if we can, just for a second, with this statistic that I've got up on the screen. Because I think this is a jumping off spot that really tells us how we are advancing our understanding, and also it's creating a fugitive, disruptive energy that is creating in our systems, both political, economic, procedural, educational, it's creating that disruptive energy, the chaos that occurs before change. Do you feel it today? Man, oh man, you can feel it big time, right? I need to look at Brexit or I need to look at Trump. Either one, I can feel it, I can tell you. So without getting too far afield into politics, let's stay on medicine. So this is an article that appeared in a very esteemed journal of the Nature Medicine Journal in 2015, written by a colleague, Nicholas Schrock of Craig Venter. Craig Venter, you probably know, was in the United States one of the people that was involved with the Human Genome Project through his company Celera, They raced the government scientists to see who could be the first to decipher the Book of Life, and there's been a debate as to who won. Some people say Greg Venter won, some people say the government scientists in Britain and in America won. Whoever, anyway, it was a competition. So Craig Venter and Schrock were very involved with this whole development of the genomic model of the gene. And this article, I think, is a very interesting article because what Schrock did is he went back and he asked the question, if you took the 10 most frequently prescribed drugs in America and asked the question, from all the research that's been done in their approval, large, huge studies, multiple thousands of people, billions of dollars spent, what's the success of these drugs in the proper use of these drugs in the treatment of people with that diagnosed condition? That seems like a reasonable question, right? So let's go back and retrospectively review. If a person has the disease for which that drug has been approved, and you gave it then to 100 of people with that disease, how many of them get a positive outcome? Now, we might assume that you're going to get a hugely high percentage of people. I mean, after all, these have been approved by the government. They are highly studied, billions of research dollars. So we would come to the assumption that they must be highly successful. So here are the 10 top drugs at the time in America that are being evaluated. And what you're looking at is the blue little humanoid figures represent, out of that number of people, how many get a positive response, and the red represent how many people get a non-response or a negative response, right? A non-successful response, let's say a non-therapeutic successful response. So you start with Abilify. You probably know that is a trade name for an antidepressant, an SSRI. So that's 1 out of 5, right? So that's 20% people get positive response. Then you go over to Nexium. That's for H2 blockers. That's for esophageal reflux disorders. And you get 1. You can count the numbers of reds there, right? So it's 1 out of 20. That's 5% of people get a positive response. Now you're probably starting to already ask the question, well then how are these drugs approved and reimbursed by the government for therapies for which the majority of the people don't get successful outcome? It's an interesting question, right? We could spend the whole rest of our time exploring that, but let me just summarize. Have you ever wondered why we have to do such huge study sizes for these clinical approval of these drugs? You might have thought it was to demonstrate safety. Well, that's a small part, but the biggest part is in order to get statistical significance in a P05 model, sometimes the clinical signal is so weak in these drugs that you have to go to a huge study size in order to get enough individuals that would fulfill the criteria of P less than 0.5 statistical significance. So it's built really somewhat on the RCT necessity, the randomized clinical control necessity to show significance as to why we have these large data sizes. Because the signals for these drugs are actually not that strong. Even with all the biology that's been, all the chemistry, all the target work that's been done, when you get to the clinic, which is where it really counts, their effects are not that good. So you can, I don't need to go through all the rest of them, but you can see there's a lot of reds, aren't there, versus blues on any one of these drugs. So you go to Crestor, that's a statin drug. By the way, if you might ask, if statins were the #1 prescribed prescription drugs for over 10 years, what disease do they treat? Right? So #1 drug prescribed for over 10 years, this family of drugs called statins. And if I ask what disease do they treat, they don't treat any disease, right? They treat the relative risk of a disease, which is called hyperlipidemia, right? Hypercholesterolemia. So when that drug was approved, I don't think a lot of docs even really thought this through, that they had moved from treatment of a disease to move to treatment of a syndrome, which is related to a number called cholesterol. The assumption was that the association of elevated cholesterol to the disease would have some pathognomonic indicator that you were preventing the disease by treating the number. And in some cases, that is true. In other cases, it's not true at all, because the etiology of cardiovascular disease had no relevance to the level of their cholesterol in their blood. So there's all sorts of stories here that I could spend time on, but the takeaway I'd like you to come away with from why I'm going on to this in some length is that this means that we have a lot of room for improvement of efficacy of intervention by personalization, because you can't just talk about a class of a drug against a class of a disease and expect everybody in that class to get a positive effect from a single molecule. Now we have 14 different drugs in the United States that are approved for treatment of type 2 diabetes. Why do you need 14 drugs? because diabetes exists with a variety of different etiological phenotypes, or what are called pathophenotypes. And those different pathophenotypes don't all respond to the same signal. One might be something that blocks glucose uptake in the kidney. Another might be GLP-1 and its relationship to signaling at the intraendocrine system. Another might be sulfonylurea that blocks certain transport processes related to ions across the cell membrane. So there's different ways that one can approach this based on the individual need of the patient. Okay, so that leads us to a different biology, and I'm not going to go into this in great detail, but I'm going to talk about two words that you may be familiar with, or two terms. And these are words we memorize. They're called disease adjacency and comorbidity. Now, if a woman were to appear who has rheumatoid arthritis, cardiovascular disease, and osteoporosis, in our system of healthcare, and I believe it would be the same here, she would likely be seen by three different subspecialists. One that would treat her rheumatological condition, immunological problem, one that would be focused on her cardiovascular, and one that would be on her bone problem, right? Endocrine related. So you'd get three different subspecialties with three sets of agents that would treat three separate diseases belonging to three different country clubs that have three different sets of colleagues and meetings they go to. All in the same body, call that patient. Now, what we call those conditions in, to make that understandable, is we call them comorbidities, because we recognize that those three conditions often do cluster together in the same person. So we call them comorbidities, or disease adjacencies. Now that makes the assumption when I use that language that they don't have any connection in actual fact at their etiological level. It just happened to be circumstantially seen, occasionally coming in the same patient. You get where I'm going? Yeah. That's an old model. That is an old model. I'm telling you, that is, the death sentence for that model has been written, maybe not totally published yet, because we're still holding on tenaciously to that concept that these are three separate diseases with no common theme. But actually, in understanding the etiology of these diseases that have emerged over the last 20 years, we see that they are connected both genomically, phenotypically, and biologically. So it's highly, and that's what these little lines in between the circles are. Each of those circles is a different disease. The size of the circle represents its frequency in the population. The lines between them represents that they're interconnected either by their genomic disturbances or by their phenotype, so that they really are not comorbidities. They suffer from the same disturbance of physiology. So maybe if you treated the cause and rather than the effect, you'd get a different outcome of all three diseases by a single intervention. You follow where I'm going here? Now this is a remarkable change, and now I'm going to give you another part of this. I'm cutting the end of my talk so you won't have to listen to the rest of the meandering. But now we recognize that for most of these chronic conditions that we see today, the dominant disease indices that share comorbidities with other conditions, that the major wild card that leads to their etiology, the major wild card happens to be related to lifestyle, environment, and diet. It's not the absence of a drug. It is the way the genes interact with those variables: diet, lifestyle, and environment. Ta-da! So where do you think the future of healthcare is going? Is it going in a multiple development of all sorts of heroically new drugs to treat every possible permutation and combination of a gene's environment interconnection? Or is it going to understand how to personalize diet, lifestyle, environment to treat cause rather than effect of age-related non-communicable diseases, which are the epidemic that are causing a ruination of healthcare economies globally? We're still chasing an old elusive model of an infectious disease with a bug to be treated by an antibiotic by trying to use that same model to treat chronic diseases that are pleiotropic, multifactorial diseases of lifestyle. And this model will never work. And the drug companies, by the way, know that. They know that they cannot develop drugs fast enough to cover the diversity of conditions that emerge out of a mismatch between genes and environment because of the variegation of genetic uniquenesses among our population, much greater than we previously understood. So this is what Barabasi is talking about in this article, that we live in these layers, these genomic layers, our metabolomic layers, and our sociogenomic layers. People hang out together that share common lifestyles. So if you have friends that are obese, it's likely you'll have a weight problem. If you have an alcohol group of hangout people that's the way they socialize, it's likely you'll have an alcohol problem. If you have friends that are all diabetic, it's likely you'll be diabetic. This social genomics ties us to our phenomics, which ties us to our genomics, right? It's a cultural web in which we live that then give rise to the dominant diseases for which then we have been chasing them, trying to find the drugs to treat them. Unsuccessfully, I might add. Okay, with that as a disturbing concept, then is there an alternative? Yes, there is, and this is emerging now as the new science that is really changing all of medical education. It's changing the whole way we view physiology and anatomy and pathology. And this is described by Albert Laszlo Barabasi and Joseph Locoso here in this recent book this year on network medicine, that it ties these together into a new model, an interconnected model where we're finding ourselves as part of this web of life. And our physiology is part of the web of understanding. And so now, let me give you the new model in summary. The new model is that we have all these antennae sitting on the surface of our cells. These antennae, I call them receptors, are receptors for vision, receptors for smell, receptors for taste, receptors for touch, receptors for electromagnetic radiation. Those receptors on every cell of every tissue of every organ system of our body are picking up information in real time, just as you are right now, as your tympanic membranes are picking up the vibrations of the air, experiencing then a neurological response. It's giving you a sensory response that's saying, I just want to leave this meeting, or no, he has something interesting to say, right? So you're getting a biobehavioral response to the movement of air that's transduced through these receptors called hearing. Similarly with sight. Similarly with touch, similarly with electromagnetic radiation, like just having come from Brisbane, I can tell you, when you come here, you know you've gone through some magnetic lines of force. We call it jet lag. Your whole circadian rhythms, I have one of these little aura rings on, and it tells me exactly how my flight influenced my biorhythms, right? So the construct that there are these receptors that are influenced by the things of daily living, what we smell, what we eat, how we act, how we think, is a dominant new view that is unique to the personalization because we each respond in different ways. If we had a loud noise in the room, suddenly unexpected, some of you would jump out of your seats and you would just be alarmed and look around for what's going on. And some of you would just sit there and say, hey, there was a loud noise, right? So we have different responses to stimuli by the way that our receptors are triggered into a neurophysiological response. So that model is a very different model of the cause of dysfunction, or let's call it functional change, than just saying a bump in the night we got heart disease. So this leads us to want to treat the cause and not just the effect. This is the basis for the personalized nutrition functional medicine model, is let's dig deeper, let's look at the root cause and try to see if we can't find it. So let me give you some examples of how this plays out in 2019. So this, I'm going to give you a quick summary of what I consider the largest study ever been done on the evaluation of how genes are influenced by an environment. So these are our space lab studies, right? Between two identical twin brothers, both medical doctors. You know this story. One stayed on land, one went for nearly a year up in Skylab. And they had everything you could imagine measured about them. All the genomic sequencing, microbiome, all stress, all neurophysiology. I mean, these were measured like max evaluation of biometrics. And what they found is that the twin that went up, oh, and by the way, they're identical twins, right? They're identical twins, so they share about as close as you can have to identical genomes. I didn't say epigenomes, I said genomes that you can imagine. So the twin up there in space, as he's being measured, his biology changes compared to the twin that's on land in Florida or in Houston. And so over time, what occurs is actually his epigenome, at which they're measuring the so-called methylation patterns of his genes, actually change. So his genes in themselves don't change, but the marks that are placed on the genes, the epigenomic marks, which regulate how the genes are expressed, change. So the book of life is the same, but certain chapters are expurgated in one twin versus the other. Get where I'm saying? So they change the way the genes are being read. So when they come back, when he comes back, he has all these things that the two brothers are having measured on them. And I'll just let you, so the biochemical cognition studies, epigenomics, gene expression, immune function, metabolomics, microbiomics, using metagenomics, proteomics, physiological stress testing, and telomere length, right? So there's a lot of stuff measured on these identical twin brothers. And what they find is that a year in orbit produces huge changes in the way the genes of the brother that's being, I guess you would either call it stressed or whatever you might want to call being in space. I think I'd call it stressed. The influence that it has on the way his body is functioning. His genes remain the same, but the way they're expressing their function is extraordinarily different. It's like they're no longer twins, right? And this happens in a matter of a very short period of time. So we could actually go through and look at all the analytes that were done, and I'm not going to bore you too much. I'll just pass through these quickly. So the telomeres change. The length of the little caps on the end of the chromosomes change. And the methylation patterns of the genome that relate to how epigenomics is regulating their gene expression change. The greens and the blues are the two brothers in all of these slides, by the way. Their response to vaccines and their lipidology, meaning their complex fatty acid profiles change, and their immune system changes. Their fecal microbiome changes dramatically over the course of this period of time. Their mitochondrial bioenergetics changes at the level of how they process energy within the cells of their body and the energy powerhouse of the cells. Their fluid shifts and their vision changes through neurophysiological changes in the way that signals are transduced as a consequence of these epigenomic changes from space. Their cognition changes, right? In fact, the brother in space, it gets dumbed down versus the brother on land. Now, the good news is that most of these things, when the brother in space comes back to land, they revert quickly back to his baseline like his twin brother. But some have not yet reverted back. So the question is, you have a very high intensity environmental change. Does that produce stickiness in your epigenome that's hard to get rid of, like post-traumatic stress syndrome, for instance? So what I've just told you is this study, which is the largest and most significant, and you might call it extreme study, looking at the effect of environment on genomic expression, shows you can take two identicals and over the course of a period of a fairly short time, by different environmental exposures, completely alter the way their phenotype looks. So, wow. So what happens then if this happens slowly over time, and we call it aging, or we call it loss of function, and we don't have an explanation how that occurs? I used to have slides in my presentation that I would show pictures of incarcerated young men and women that were available on the internet from frequent arrests for drug abuse. And I stopped putting it in there because the slides are so dramatic that for some people they said they really were discouraging. So you would have, say, a photograph of a woman the first time she was incarcerated, say in her mid-20s for drug abuse. And then between 5 and 7 years later, there would be other arrests, and you would visually see the change. And literally, in looking at these patterns of 30 or 40 different people over the course of 5 years, you would see that not only did their whole physiognomy change, they aged dramatically, rapidly, but if you look in their eyes, it's as if their soul was stolen from them. It was no one at home. Now, this is a small molecule, right? Think of this. Drugs are small molecules that are certain drugs are so powerful in influencing receptors on our nervous system, they actually can steal from us the most private thing we own, which is our soul. Just let that sink in for a moment. So this construct that we are in a fluid relationship all the time with our environment, and whatever these genes were that we got through Mendel's laws of recessives and dominants are all kind of modifiable in their expression based upon our experience of life and where we select or how we selected to live, or maybe how we did live. Maybe we didn't even select it. We just got thrown the lot of the life, and that influenced how our genes are expressed. Now the good news is, as was shown in these twin studies, is that when we get back onto land, meaning home base, home meaning safety, we can have our genes restore genetic function, and or original genetic function. And I've been studying quite a bit recently what is being termed the science of goodness. The science of goodness is a very interesting concept, right? Because what we all recognize, everybody in this room, is that the dominant concerns we have in our health patterns globally now are called inflammatory diseases. Every one of the major chronic age-related diseases has an inflammatory component. Now, what is inflammation? It's the body's natural reaction to being at risk, right? We need inflammation. It activates our immune system to defend against a foreigner or an injury or recycle damaged tissue. It's an important process. Without inflammation, we'd be subject to opportunistic infection and all sorts of things that would limit our life opportunities. However, just like any good thing, if it's overused and it becomes pedal to the metal as a chronic metabolic inflammation process that lives with us 24/7, 365, now that good thing turns against us and it becomes rubor, color, and dolor. It becomes the god of war, color red, called Mars, right? It becomes the diseases of arthritis, of cardiovascular disease, of dementia, of cancer. All these diseases have an inflammatory component. Now, why is that? It's because the other genes in our body, which regulate what I call bliss, which regulate balance against the hostility that we experience. So we're in constant yin and yang balance, aren't we, with our environment? One thing activates our goodness, another activates our alarm. So if everything is activating our alarm and little is activating our goodness, what's that look like in the patient? We see it. It's the diseases of today. And so the construct is, let me use an example. If I said oxytocin, what would you say? You would say, oh, pregnancy, right? Oh, baby delivery. Oh, euphoria. I mean, a woman who has experienced child delivery will tell you go through this extraordinary pain, but then this oxytocin rush you get is the most remarkable thing that actually allows you to have another baby after you've gone through that pain the first time. Right? If you didn't have that, it would be like the science of badness. And so, you know, the science of goodness is an extraordinary rush of euphoria. Now, men have oxytocin as well, right? They don't experience it quite maybe as dramatically as a woman goes through childbirth. But oxytocin is one of the neurohormones that regulates goodness, regulates our feeling of goodness, of empathy. Now, think about that just for a moment. This is the 40th anniversary of the book written by the Oxford scholar called The Selfish Gene. You probably are familiar with this. That book caused a revolutionary debate about why we as animals exist. And in that book, the hypothesis was that we exist to allow our genes to replicate themselves. The selfish gene. It doesn't care what we do with our lives. As long as we replicate the gene, we are just there as vehicles for genes to be replicated. In all plants and all animals, the same thing. They're just devices for gene replication. That's a very unromantic way to think about life, isn't it? And there are big debates about that, and people have fought back very significantly, I being one, because I don't believe that that is true. Because if it were true, there would be no reason for a person in their post-reproductive years for worried about posterity. If we're just here for a replication of our genes, by the time we got over replication age, we say, you know, whatever is good for me, that's fine. I don't care about posterity. I've already done my job. And so the construct of empathy, of cooperation, of looking for legacy that leads to survival of people that will be here when we're gone, that's something beyond just the selfish gene. That's related to the science of goodness. Because the science of goodness, these genes like vasopressin and oxytocin and others are conditions that create in us the feeling that we have responsibility for others. and that we are in service. We're not just here to replicate. And so the biggest maybe responsibility we have is not just to produce a progeny, it's to be here to create social service and survivability of the species. So this model is a different genetic model. It's one that leads to cooperation rather than survival of the fittest. And medicine, I think, unfortunately received, without choice, this Mendelian Darwinian model of natural selection and deterministic genes. Think of that if you would. Those two concepts, which were born out of Europe, right, in the 19th century, Darwinian natural selection, by the way, if you read Darwin, you'll find that we've over-socialized what he really said. He didn't actually say that, but we've socialized that. Survival of the fittest and deterministic genes, dominant and recessive, so that nothing you can do about your genes. If you got the bad luck of the draw, so as you didn't even fill out an application card, tough break, right? And so medicine is there to save you from bad genes and to help you be able to survive against the fittest. That's a very interesting model of a disease care system. I would say that's not a model of a healthcare system. Do you hear what I just said? That's a very interesting model of a disease care system, not of a healthcare system. And so the other model is the science of goodness model. It says we've got this yin and yang going on all the time between defending against the things that are trying to take us, like infectious disease, and the other, which is our altruistic ability to want to cooperate and have empathy and create survival over the long term, not just for us, but for the society in which we live. That kind of interesting interrelationship gives rise to a different model of how we would see people in the social milieu that we call a disease-focused society. Because disease is very negative. How many people really want to hang out with a fear of disease? It's like, oh boy, let's spend the next 2 months really thinking about disease. You know, wouldn't you really prefer to hang out thinking about how good your genes could be? And so for me, the single principle that I have, that I believe our genes are there to allow us to express our full goodness. Take that on for a second. That what our genes are there to do is to allow us to express our unique, full capability of goodness throughout however many years we have the fortune of living on this planet. Not that we have disease genes. We have disease for function that give us the ability to create goodness. Wow. That's a pretty interesting way of thinking about this. So that means every time you eat, you want to eat good food that leads to a good planet, that leads to posterity and cooperation, right? So now we're connected to the soil. We're part of a system. It's all interconnectedness. It's not like separate from and survival of the fittest. At your expense, I win, you lose. That's a whole different model, right? Now the fortune, I believe, when I grew up and was a medical school professor for 13 years, the majority of my students were men. That was the way it was, 30 plus years ago. Now in American medical schools, we have more women in medical school than we have men. And we are starting to see the essence of cooperation, which I think is more dominant in the female of the species than the males, to start to recolor medicine. This may be our savior, by the way. This is the age of the feminine, right? This may be the savior. Because with that, men have had a chance to do their stuff, which are really good at piece parts and mechanics and kind of taking big things apart and putting them back together. Really good at that. Men are not so good in cooperation. They're not so good at systems thinking as it relates to the things I'm talking about. Those are skills that women have, and maybe for reasons of biology, I don't know, that pertain to child rearing and so forth. But all I know is this is going to be a very interesting transition culturally. I think as we watch how this energy travels through society, moving away from survival of the fittest to cooperative survival of the species, which is an interesting thought. Okay, I just went through a big philosophy course. I apologize, but you were very kind to allow me to step off the edge there. So this concept of nutrition, where personalization of nutrition has come from, had a long, interesting history. And I'm going to just spend a few minutes talking about where it's come from, starting back really in the United States in 1909 with the Wiley Food and Drug Act, the first person who, Wiley's raiders, who said we were poisoning people with bad food because it was adulterated food. So the FDA, the Food and Drug Administration, was formed. And from that, then, this remarkable book, which was one of the first best-selling books in America on nutrition, was published in 1933. And this is, the title of the book is self-evident, The 100 Million Guinea Pigs, Danger in Everyday Foods, Drugs, and Cosmetics. And who were the 100 million guinea pigs? They were people living in the United States at the time. Because what these authors were saying, which were prominent guys in the establishment of the Food and Drug Administration, is that there was such adulteration occurring with our food supply and putting chemicals in it and starting to make processed foods that it was with no oversight and no regulation, that it was really harming people's health. So this goes all the way back in our country to the turn of the last century. Arthur Callett and Fred Schlink were the two principals. I won't go through them, but that they were very interesting figures in history, and they had this good friend who turned out to, Ruth DeForest Lamb was her name, and this woman was the Food and Drug Administration's first chief educational officer that was there to educate about foods in the United States, and she wrote a book following on their book called American Chamber of Horrors. And what do you think she was talking about? The food supply. That as an FDA education officer in our government, she had a best-selling book saying, hey, by the way, this food that we're starting to see for the first time, this shelf-stable processed food, is a laboratory of horrors. We don't even know how safe this stuff is. It's taking us in a different direction. Well, that's all. I can't say any more. I've lost my slides. I'm sorry. So, you know, only get one shot, and then they pull the plug once I get too political. So it's not the first time this has happened in my professional life, I can tell you. So the upshot of all of this is that we go through now into the next epic period with another person whose name you might be familiar with because they had impact in Europe as well in the United States. I was raised with this person. She was a dietitian, master's in biochemistry from the University of Southern California. And I'm just going to pass through quickly. And her name is Adele Davis. And my mother was an Adele Davis devotee. We were raised around Adele Davis nutrition. She had the first 20 million book sales in nutrition in the history of the English language. And her books were really, she had actually 35 years of clinical experience as a nutrition expert managing patients, but did research. Her first book sold over 10 million copies, and really a remarkable person. And she actually was the person who was the predecessor, if you look back in 1927, to two other name figures that are very important, Linus Pauling and Roger Williams. And Roger Williams and Linus Pauling, as you probably know, the book that started the nutrition supplement industry was Linus Pauling's book entitled Vitamin C and the Common Cold and Flu. Everybody now knows about vitamin C and the common cold. It really came about from this book. And the reason that he became so popular, he was a two-time Nobel Prize winner. So here's the first time that a major scientist was publishing things about nutrition and vitamins and its influence on health. So all this kind of occurred in the middle 1900s. Henry Beeler, then a very prominent physician in the United States, wrote Food is Your Best Medicine. And then later, the head of biochemistry at the University of California, Berkeley, one of the most prestigious universities in the United States, Bruce Ames, started studying the role of vitamins on health, and he found out that there's a lot more to vitamins than just the prevention of scurvy, beriberi, pellagra, xerophthalmia, and rickets, the deficiency diseases, that actually these vitamins have different effects on function of individuals based upon their genetic uniqueness. And the range of need for optimal function may be much greater than the needs to prevent scurvy or beriberi or pellagra. So this was the birthing of the science of the nutrient supplementation era during this period of time. So that then leads to the development of what we call scientific wellness now as we move into the 21st century. And this is how we measure all these different things in people and ask what is the optimal nutrition and health support for a person, not a community, not a population, but an individual. And I was involved in this pilot study that was 5 years ago called the Pioneer 100 study with 108 individuals, had over 3 million data points taken of their biology over the course of 3 years. And so what we saw was the acceleration in the development of personalized nutrition from this data set. And so the whole nature of nutrition is going from epidemiology to personalized nutrition. A lot of what we've been applying is epidemiological statistical nutrition. Now we're looking at individual nutrition. So Science Magazine, one of the reputed, top-level journals in the world, has a whole issue on diet and health related to personalization of nutrition in November of last year, in which they talk about personalized nutrition as a contributor to human function, how we move from being average to move to being optimal. And we look at dietary fat in a new way, saying maybe it's not the villain that we thought it is. Maybe actually dietary fat is good for us if it's the right type and the right amount. It's not to try to cut out all the fat of the diet and waste away into a desiccated ball of bones, that really maybe the right kind of fat in the diet is good. And in fact, if you look at the relationship of dietary fat to mortality, you find people that increase their omega-3 fatty intake have the lowest cardiovascular mortality. So increasing the right kind of fat has a positive benefit. And we now know that highly processed carbohydrates, simple carbohydrates, are associated with increased mortality of virtually every chronic disease. So it's the type of macronutrients, not just the amount, that leads to the alteration in physiological function. And in fact, this article talked about carbotoxicity, that when people are eating too much of the processed carbohydrates, and this was actually in one of the top basic science journals called Cell, an article on carbotoxicity, noxious effects of simple overly processed carbohydrates. Who would have believed it, that at the highest level of cellular biology academically we'd start seeing these articles? So there are points of consensus about how these diet principles influence function. Focus on nutrient quality. A reduction in chronic disease can be achieved by people with diets with a broad range of carbohydrate-fat ratios if those are minimally processed and in a natural state. Replacing saturated by unsaturated fat is desirable, particularly the omega-3 fats. This is now a commonly held concept. Removal of highly processed carbohydrates and sugars, where we used to hear that sugar was just a good energy food. Now we're saying, yeah, it's a good energy food for early diabetes, cardiovascular disease, and Alzheimer's dementia. Personalized response to diets is now being recognized universally. Ketogenic diets may provide value in certain individuals, not just those with seizure disorders. Well-formulated, lower-carb, higher-fat diets do not require intake of excessive proteins or animal products. You can be a vegetarian and eat healthily. And nutrigenomic science is opening the door to personalization of the diet based upon genetics and nutrition. And so we now see that there are really good studies in top-rated journals like the New England Journal of Medicine showing the benefit of omega-3 fatty acid supplementation in the reduction of things like cardiovascular disease and diabetes. We recognize that fasting physiology is starting to become a major important new theme with time-restricted feeding. I was just at a meeting on the way out here in Australia with Sasha Nandapanda, who you probably know is now one of the top chronobiology investigators at Salk Institute in California, and talking about the 18-6 program, which I have been living at myself. That means you have 6 hours of, well, it could actually, it's the 16-8 is probably the more applicable program. That means 8 hours of eating throughout the day, 16 hours of non-eating. So you can figure out the timing. For me, it means stop eating around 7 o'clock at night and not eating until around 11 o'clock in the morning. So that gives you a mimicking of a fasting physiology and an implemented daily regime that doesn't put you at great disadvantage. You can still have a social life. You just stop eating at 7 o'clock at night. You don't have big late meals, which for a lot of European culture means you better adjust the wheel to a different time. You want to get that space of 16 hours of non-eating, sometimes throughout the day. So increasing interest in fasting physiology, ways of introducing fasting physiology with time-restricted feeding or fasting mimicking diets, which I've been heavily involved with Walter Longo's studies at USC on the fasting mimicking diet. So we're starting to see diet become therapeutically an important part of health in ways that are personalized to the individual's need. And how that relates to their circadian rhythms and their clock genes and this whole nature of rhythms of life, rhythms of hormone, rhythms of eating. Every cell has its own biological clock that is controlled in part by the central clock in the suprachiasmatic nucleus of our brain, which is called the BMAL and clock genes, that regulates how things are being processed. So you want to eat at the right times, and you want to take drugs at the right time, and you want to make sure your fluid intake is correct for hydration. All these things are becoming absolutely new science that is driving personalization and producing improved function. And then we lastly put onto that the microbiome, right? This new revolution that you're going to hear much more about over the next part of today from the experts on the speaker roster. This microbiome diet connection is a fascinating new chapter that we're going to be able to harness much more effectively over the years to come. And I'll just say that we were very fortunate now about 10 years ago to develop a relationship with a group out of Louvain University in Belgium, the Catholic University of Louvain, originally it was with the senior lecturer on gastrointestinal function there, who now is a professor emeritus, and his postdocs took over his laboratory, Patrice Connie and Nathalie Delzen. And they are producing extraordinary data on the role of microbiome on stabilizing insulin, causing weight loss. That the gut bacteria actually have something to do with our body weight and our fat deposition, about relationships to certain species of bacteria in our gut having to do with stabilization of our immune system, reducing inflammation. So we're now starting to see that harnessing this extraordinary complexity of our gut microbiome, which is related to not just what we eat, but how we look, act, and feel, the stress we're under. All of these things are part of a universal new system that allows us to start fingerprinting how we personalize lifestyle. And that's why our icon for the personalized lifestyle medicine is a fingerprint, because we're moving from the age of populations to the age of individuals. And as we do so, we will beat back against the rising tide of chronic disease that is not diseases of populations, it's diseases of individual. And health starts one individual at a time. So with that, I thank you for your patience and allowing me to wax philosophically, and we'll see where we go from here.